something at the beginning.
So today we're going to have a guest lecturer. Gadi Getz is the Director of Bioinformatics at MGH and also the Director of Cancer Genome Atlas at the Broad Institute. He's also a professor of pathology at Harvard Medical School. He is the world expert in tumor genome analysis. And welcome, Gadi.
Thank you, Shirley, for the invitation. It's great to present in this course. And I know this is a remote teaching, so please feel free to ask questions when you need to ask questions. So first of all, a bit of an ad. I'm looking for master's, PhD students, and postdocs. So if you're interested, please send me an email. So cancer is a disease of the genome and the epigenome. We all start from a fertilized egg with potential germline risk alleles for developing cancer. Then as cells divide, they accumulate DNA damage. A lot of the damage is repaired by repair mechanisms, but some of the damage is not repaired and they stay as mutations in the genome. Most of the mutations don't do anything or maybe even are slightly deleterious to the cells, but some mutations could increase the fitness of the cells and the cells start to either grow faster or die slower and accumulate and creating a clone. These clones are not yet cancer, they are pre-cancer, and they continue to grow, and at some point additional driver event could happen, and that could eventually transform into cancer. At some point there could be that the immune system identifies the clone and starts to attack it, and there could be driver events that evade the immune response. So all these mutations that increase the fitness of the cells, we call them drivers, and all the other mutations that are happening and don't increase the fitness of cells, we call them passengers. Eventually, when cancer is diagnosed, the cells are highly rearranged with many mutations, could be between 0.1 to 100 mutations per megabase of the genome, and could be thousands of structural variations, but only roughly a handful of them are actually drivers that gave fitness advantage to the cells. Here's the last clonal driver, meaning it's all the cancer cells are descendant of this cell, and they therefore contain all the driver and passenger events that happened all the way back all to the fertilized egg. And here are subclonal drivers. So for example, this set of cells are descendant of this cell, and they are now, let's say, 20% of the cancer. Then there's treatment, and treatment could kill many cells, but maybe some of them happen to harbor a mutation that makes them resistant to the cancer, and then the clone continues to grow, and eventually there's a recurrence, and which continues to evolve, and maybe additional drivers are accumulated during this recurrent stage, and then a different treatment is given, and then again another resistance could occur, and this is kind of the life history of a cancer, at least ones that are not cured. These mutations are caused by different mutational processes. There are natural mutational processes that occur of every cell in the body, like these CpG to T, methylated cytosines in CpGs that transform to T, and this occurs throughout life. And there could be other signatures that are caused by external kind of carcinogens like smoke, cigarette smoke, and those could have impact and generate their own mutations with their own signature. And there could be other types of processes that, for example, one of the driver events could be losing some DNA repair, and from that point on, the cells have more mutations because they are less repairing mutations or damage that occurs during cell division. So just to summarize, drivers can be genetic or epigenetic, anything that goes from mother cell to daughter cells. They could be in the coding regions, could be non-coding regions. They're generated by different mutational processes, and they together kind of aspire to achieve the cancer hallmark, like evading immune response or uncontrolled growth, etc. They could support becoming resistant to cancers. They deactivate or activate different molecular programs. Cancer is heterogeneous, as you can see, and some of the mutations could expose germline risk allele. For example, the famous BRCA1 or 2, a person could have one good copy and one bad copy of the gene, And if they lose during life the good copy, then the cells are left only with a bad copy and therefore have less of repair. So cancer is a dynamic and heterogeneous system, which is a kind of all the ingredients that you need for Darwinian evolution within the body. So why are we interested in all this cancer and drivers and the evolution of cancer? Is because of targeted therapies. Here is kind of a poster child example of a 38-year-old man with metastatic melanoma that happened to have a BRAF V600E mutation. And this was at the time where vemurafenib was still kind of experimental. This patient got vemurafenib after a few weeks. There's no sign for the cancer. The sad part is that few weeks later, the cancer became resistant and it came back. But this is kind of a typical trend now, with moving from more anatomy-based treatment like we used to do in the past to more genetic or molecular-based treatment, where cancers are being treated with these targeted therapies that are addressing those particular proteins that are being altered or kind of essential for the cancer cells. And there's a growing arsenal of these targeted therapies. I think for cancer around the 85 or something like that now. And for example, recently there was FDA recently approved the FGFR inhibitor for bladder cancer. So in order to understand the cancer genome, there's two major tasks. One I call characterization, which is to really understand in an individual patient what happened from the normal cells to the primary tumor to the metastases. So comparing their germline DNA to the somatic changes in the tumor and the metastases. And then the interpretation task is to taking all these characterization data across a cohort of patients and then ask what are the genes or the parts of the genome or the pathways that are altered more than you'd expect by chance, to identify what are those drivers compared to passengers, to identify subtypes of the disease, to associate with clinical parameters. In terms of the statistics or statistical power in the characterization part, the power comes from, for example, depth of sequencing and how many samples you have for that patient. This tells you how accurate you could characterize what happened to the cancer from the normal to the cancer cell. How, if you're doing whole genomes versus whole exome, all of this will give you power to detect these events. In terms of the interpretation, the statistical power comes from the number of patients. The more patients you have more power to detect these drivers or subtypes and associate with clinical parameters. So all of this field was kind of transformed by this kind of graph that you've probably seen many times, which is the drastic kind of exponential decrease in cost of sequencing. Going back from 2001 with $100 million per genome sequencing to $1,000 today. And that is equivalent for a Tesla costing like $35,000 roughly to costing 35 cents. So this is a huge drop in cost that enables generating a large amount of exomes and whole genomes on cancer patients, which gives us both the ability to characterize their genomes and to interpret and identify the drivers. Addition technologies like this capture technology that enables capturing parts of the genome, for example, the exomes, enables even a cheaper sequencing of exomes, and then therefore doing more of them. Since the cost of genomes is dropping so dramatically, at some point it might not make sense to do the exomes anymore. We'll just do the entire genomes. So historically, since 2006, started the TCGA, which is the national project to characterize cancer genomes, and then extended in 2009, and essentially ended in 2016 with a paper still published later from the consortium. And this is a kind of the Cancer Genome Atlas with the aim of studying 20 to 25 tumor types. Eventually studied 33 tumor types with some smaller cohorts with rare tumor types. every one of these large tumor types studied 500 tumor normal pairs with the whole exome sequencing to all of them. 10% had whole genome sequencing. Now there's more whole genome sequencing being generated. 20% had low pass whole genome sequencing, all of them in the RNA-seq for from mRNA and microRNA, SNP arrays for copy number and germline kind of SNPs, methylation arrays, and the proteomics done by reverse phase protein arrays. The International Cancer Genome Consortium is an umbrella organization for similar projects around the world. And the goal of the ICGC is to study 50 tumor types and the subtypes. And recently, just earlier this year, there was many papers published out of a collaboration between TCGA and the ICGC to study whole genomes called the pan-cancer analysis of whole genomes. The primary goal of these projects is to basically generate a comprehensive catalog of all the cancer genes and pathways and subtypes, so we will have potential targets to develop drugs. So I will stop here for a second.
We have questions? Okay.
So now I would like to describe the efforts in characterization. So again, the goal of the characterization is to reach a complete, hopefully base level characterization of every tumor within the body of a patient. And that, of course, we would like to do it at every cell in the body of the patient. That, of course, is still not feasible, but that would be kind of the complete characterization, fully understanding what happens in the genome and epigenome of every cell in the cancer. So the solution is, of course, massively parallel sequencing, also known as next generation sequencing, that enables base level resolution of the genome. There are many challenges due to that we have been addressing in the past decade or so, and I will kind of give you a few examples of those. The first one is the flood of data. Because of this drop in sequencing costs, the amount of data is increasing dramatically. We've started developing in 2009 a system at the Broad Institute called Firehose to kind of analyze the world's data of cancer genomics. This at some point, the computers in the Broad Institute were not strong and large enough to handle all the data. So there was an effort together with the NCI also, to move these analysis pipelines to the cloud and build an infrastructure to run pipelines on the cloud. And we've built FireCloud, which is now kind of even further expanded to a system called Terra, and you're welcome to use them. And these are the URLs. And a lot of the data sets that are generated are available there. And also many of the tools and pipelines you could find them and run in the cloud. So in order to do next generation sequencing base level characterization, here's a cartoon of what we're doing. And as you've learned previously in the course, you could take DNA fragments, map them to the genome, and then we could identify point mutations by looking at kind of sites in the genome where the base in the cancer does not match the normal cells of that patient. We could identify point mutations and indels. We could identify copy number changes by the depth of sequencing. We could identify translocations or structural variations by looking at these pairs of reads and seeing that one part of the read goes to one place in the genome and another goes to a different place of the genome. And that helps us identify translocations and structural variations. And we could even pinpoint the exact breakpoint to the base level resolution. And there are also methods to identify non-human sequences like viruses that cause cancers by depleting known human reads and seeing what's left and then identifying potential pathogens. And over the years, we've developed many tools to addressing each one of these questions. And I will give you some flavor of how these tools work and what they do.
Hi, I have a question.
Sure.
How do you, going back, how do you distinguish what is a cancerous point mutation versus what is just a normal, like a variant or a SNP that you can find across the population?
I will discuss this in a few slides, so we'll get back to that soon. So first I want to talk about sensitivity and specificity for finding mutations. So one thing that is different from the germline, that people study, take a blood sample and extract DNA from white blood cells, is that a cancer sample is a mixture of typically a mixture of tumor cells and normal cells. When you take a biopsy and you extract the DNA, it's a mixture of normal cells and tumor cells. So this poses some complication for an analysis. What are the complications? What's the effect of this mixture of tumor cells and normal cells in the tube? Is that we need to assess what fraction of the DNA in the tube came from the tumor cells. So in order to do that, we need essentially two numbers. What's the purity? What fraction of cells were tumor cells? And what's the ploity, which is what's the average DNA content in a tumor cell compared to a normal cell. So for example, if you have high purity, then of course you have more DNA coming from the tumor. If you have also high ploidy, you'll have more DNA coming from the tumor, higher fraction of the DNA coming from the tumor. So here's a cartoon of a cancer sample that has 2/3 cancer cells and 1/3 normal cells. And in this region of the genome, there are 4 copies in the genome in the cancer cell, but 2 copies in the normal cell. And here's a mutation, a clonal mutation shared by all the cancer cells in one of those copies of the four alleles. So what will be the expected allele fraction of this mutation? We have 2/3 is the purity. The absolute copy number is 4 here. The mutation multiplicity, meaning how many copies of the mutation exist in every cancer cell, is 1. So overall, the allele fraction would be here 20%. And the reason is you could count 2 alleles are mutated out of altogether 10 alleles here. 2 here and 4 plus 4 is 10. And 2 out of 10 is 20%. So the allele fraction of the mutation, if you sequence deep enough, you'd expect 20% of the reads to have the mutation and 80% of the reads not to have the mutation. So we could calculate a simple formula and say the allele fraction or expected allele fraction of a mutation is the multiplicity in the cancer cells times the purity of the cancer divided by the purity times the local copy number, which would be 4 here, times 1 minus the purity, which is the normal cells, times 2, because there are 2 alleles coming from normal cells. That's of course assuming we're talking about kind of not kind of X and Y chromosomes, the autosomes. What happens if the mutation is only in the part of the cells, let's say in half of the cells? So here in this cartoon, imagine that only half of the cells have the mutation. Of course, this would also affect the allele fraction because now only 1 allele out of the 10 have a mutation. So it would be 10% allele fraction. So if you sequence deep enough, you'll find on average 10 out of the reads will have the mutation. So the actual formula now has the CCF here, the cancer cell fraction in the formula. So if you want to calculate an allele fraction of a mutation, you need to take the CCF times the multiplicity times the purity divided by this same denominator, and you'll get the expected allele fraction of the mutation. When we run an algorithm to find somatic mutations, it's essentially running a classifier for every position in the genome. And like every classifier, it has false positives and false negatives. And we could characterize the quality of the classifier using ROC curves. And as you know, this is kind of the sensitivity as a function of the 1 minus the specificity or the false positive rate. In our tools, there are many parameters that affect this ROC curve. Of course, the allele fraction of the mutation would affect. The lower the allele fraction, would be more difficult to find the mutations. The coverage, the depth of sequencing, the higher the coverage will give us more power to detect the mutation. And of course, the noise level in the system, the higher the noise, will have less ability to find mutations. So now I want to give you a bit of an example of MuTect, which is the algorithm that we've developed and we still use until today to analyze and find somatic mutations in cancer. And I'll get back to the question about tumor and normal in a second. So first, the sensitivity to find mutations. So the frequency of single nucleotide variations is roughly 1 mutation per megabase in the genome. Of course, it varies quite a bit, but just to give you a sense, it's 1 in a million bases is actually mutated in the cancer, at least clonally in all the cancer cells. So let's imagine an experiment where you sequence this sample that has a mutation that is, in this case, it's a subclonal mutation with an allele fraction of 0.1, and you sequence it to a depth of 30x. So every part of the genome is covered by 30 reads on average. So let's say this site with a mutation is covered by exactly 30 reads. How many reads do we expect that will have the mutation? This is just a binomial distribution with a p of the coin at 0.1. So there's some probability, 4.2%, that we won't get any read with the mutation. And there's some probability of 14% to get 1 read and 2 reads and 3 reads, etc. The expected value is, of course, 3 because it's 10% out of 30x. So the theoretical sensitivity of a mutation caller, any algorithm, the theoretical sensitivity cannot exceed, in this case, 96%, because in 4% of the cases, there are no reads with the mutation. So of course, no algorithm will be able to figure out that there's a mutation if there are no reads with the mutation. And if you have a very kind of easy, excitable algorithm that even with a single read with a mutation will call a variant, then you will be able to find it in at least 95% of the cases, have more or one of the reads. And then if you need 2 or more reads at 80%, and you need 3 or more reads, your sensitivity will be 60%. So now comes the how we distinguish somatic from germline. So you've learned about IGV, I think, and this is a snapshot from IGV where we look at the position in the genome. And here you could see a pileup of reads that have this this mutation here, this blue mutation, where in the normal there's none. So this, when you look at it, you say, oh, maybe it's a mutation. In fact, in this case, it's not a mutation. It's just a sequencing noise that happened to be here, but in the tumor, but not in the normal. And this is one of the type of false positives that we have. When we compare the tumor to normal, the normal is clean. We don't see any mutation and any variant there in none of the reads, let's say. And in the tumor, we see in a few reads, but this could be just due to sequencing error. There's another type of mistake that we could make, is when this site is actually a germline SNP. And for some reason, the normal was not sequenced to a high enough coverage. So we saw only one read representing this heterozygous SNP here in the site. But in the tumor, we see many reads with heterozygous, with this variant. So we say, oh, this seems to be a somatic mutation because we see so many reads in the tumor. There's nearly only one read in the normal. Maybe that's just noise in the normal. And therefore, we make a mistake. We call it a somatic mutation, but in fact, it's a germline SNP that just happens to be hidden in the normal. So these type of mistakes are less common in the genome because they're just less germline variations in the genome compared to places where you could have sequencing errors. But we need definitely to measure these two types of errors and distinguish when we look at this type at sites that are at germline variations, at dbSNP sites, versus at sites that are not in germline variation sites. Typically, because we sequence the normal deep enough, we rarely make this mistake. As long as we make sure that the normal is covered deep enough, we rarely make the mistake of it's actually a germline. Most of our mistakes are in this sequencing artifacts. So let's kind of dig deep a bit and get some intuition about that. I think you've learned about sequencing and the quality scores that are coming out of in addition to the base that is sequenced, there's also a quality of that base representing what's the likelihood that this base is a mistake. Typically, we see qualities around the 30 or even a bit slightly higher than 30 when we sequence samples. So that means that 1 every 1,000 bases is a mistake. So if there was an A there, it could be either G, C, or T, each one with roughly, let's say, 1 over 3,000. If we assume they're equally likely, the different mistakes, which are typically they're not, but if we assume that, it's roughly 1 over 3,000 to each one of these mistakes. Now let's assume that every read has independent errors, which is a wrong assumption, but for now let's assume that. And we sequence to a depth of 30x. What will happen? 97% of the sites, if you look at them at 30x, will have no variant in them, no error, no sequencing error. But 3% of the sites in the genome will have 1 error. So if you had a mutation caller that would call even with one non-reference base, will say there's a somatic mutation here, you will call mutations in 3% of the genome. And we know that cancer is mutated in 1 in a million. So you'll definitely be swamped with false positives. Now the probability to have two errors that are going to the same base, so the A goes to G in both of them, is 0.00014, which is still more than the 1 in a million mutations per megabase. So even if your mutation caller would be excited by two reads that support the same mutation, you will be swamped by false positives. With 3 or more that are the same, going to the same allele, then you're starting to be lower, just slightly lower than the expected mutation rate. So your false positive rate is kind of roughly half of the rate of the somatic mutation that you expect. So that shows you the intuition why we need typically 3 or more reads in order to call a somatic mutation in a 30x coverage. If you sequence to 30 to 1,000x, then these numbers would change and you'll need more to be confident that this is not coming out from noise. So if I go a bit to the math that is in MUTECT we are doing this log likelihood ratio test, essentially, or testing this LOD score, comparing 2 models. One, that the site is mutated in the tumor with some mutation M with allele frequency of F, but in the normal, there's no mutation. So the allele fraction is 0 in the normal. So how do we calculate the likelihood? The likelihood is the probability of observing the data given the parameters, the errors of every base, what is the reference base, what's the mutant base, and what's the allele fraction. And if we assume the reads are independent, we could break this to this product of probabilities. And then each one of these, we could fill using this kind of formula. If the base is the reference base, then when we see in a read a reference base, it could be that it came from the mutant, but the mutant had a mistake to the reference. So this is the error divided by 3, again, assuming that all the three different mistakes are equally likely. Or the base was from a non-mutant 1 minus F and there was no error, so 1 minus E. So these are 2 options of getting in a read a reference base. Getting in a read a mutant base could be that it's truly a mutation coming from a mutation and there was no sequencing error in the mutation, or there was no mutation in that read, in that molecule that was sequenced, and there was a sequencing error that made it look like the mutation. And for all other bases that are not the reference or the mutant allele, it's just the probability of making an error there. So we use this and we calculate this likelihood for the model of particular for particular frequency. And the frequency we use is the observed frequency of we approximate the maximum likelihood frequency by taking the observed frequency of non-reference bases in the pilot. So then we could calculate the LOD score, where we're comparing the mutant version of the likelihood times the prior of being mutated, and we compare to a model where there's no mutation there, and then times the prior of it not being mutated. And if this is greater than some threshold, we call it a mutation. The threshold we use is for this delta is T, meaning that we want at least to have twice as much likelihood of being mutated than not mutated. If you plug in the priors, given that mutations are roughly in, on the order of, I don't know, 3 mutations per megabase in this case, and in this assumption, we end up with this threshold in this log scale of roughly 6.3. And if the log score is greater than 6.3, we call it mutated. If it's less, we call it not mutated. But this is not enough. After this log score, we have different additional filters, which are probably representing the fact that the statistical model is not accurate, and we're working now to even improving this statistical model. There's a question here. Yes.
Yeah, so sorry, can you go back to the previous slides with
Sure.
those formulas?
No, that's going forward. Yes, please.
Yeah, sorry, I feel like the probability doesn't add up to 1. I don't know.
The probability adds to 1. So this one, this plus this, 1 minus f times this sum f is just e over 3. So from this and this, you get e over 3. From this times this, you get 1 minus e. So you get e over 3 from here and 1 minus e from here. And there are 2 other options because there's reference and mutate and then 2 other bases.
Okay. 2
and
other
this,
options.
and this is a 2/3 and it adds up to 1.
Yeah. Okay.
Okay, great. Thank you for checking this. This is a good check. So now also the assumptions of the fact that the reads are independent is incorrect, because noise is correlated between genes, and therefore we are working on improving the method. One of the ways that we found that is improving the methods significantly is having a panel of normals. And the panel of normals is basically having many different normal samples sequence, and we look at them at that site to estimate kind of what's the noise in that site, and therefore having this filter. And we have 2 types of these panel of normals. I won't go into that, but these are dramatically improving our specificity. So how could we measure our specificity and sensitivity to generate this ROC curve to measure the quality of the mutation caller? We have this experiment that we did. We took sequencing of NA12878, which is a known normal sample that is sequenced many times, and we sequenced it twice, the same sample twice. It's not a cancer, it's a normal. And one of them we call it the normal, and one of them we call it a cancer. If we try to cause somatic mutations by comparing this tumor to this normal, then we would find all our false positives, because there shouldn't be any mutation found. We compare the same sample to itself. It's just differences in the sequencing libraries, etc. It's not a true somatic mutation. So we measure the specificity. For the sensitivity, we take a related sample. It's the daughter or the parent of this sample. So where we have SNPs in the genome, where this sample is different from this SNP, a heterozygous SNP, that this is different from this one. And we basically spike in reads from this sample, replacing reads from this original sample here at whatever allele fraction we want. So we could control the allele fraction, and then we could measure. Now we know where the mutations are because we know exactly where the SNPs are here. And then we compare this virtual tumor to this virtual normal, and then we see, can we detect the mutations? We could do this by simulation. We could also do it analytically by kind of modeling it. And this is what we came out. For different allele fraction, we could see the different sensitivity curves depending on the sequencing coverage. If you sequence to 30x and the allele fraction was 0.05, there's a very small chance you'll find the mutation. If it was 0.1, there's a 60% that you'll find the mutation. If it was 0.4, there's a very high chance that you'll find the mutation. These little dots here are from this simulation that I just showed you before with the spiking in of different reads. The solid line is from a kind of theoretical curve calculation, which fits quite nicely to the simulation data. When we did this, we compared different mutation caller at that point, and looking at the same level of false positive rate, which is what we want to be kind of way below the the 1 mutation per megabase, we saw that MUTECT was the best, had the best kind of sensitivity for this same level of false positive rate. So we showed that our method is much more sensitive than all other methods that were available kind of at that time. And other tools are improving, and our tool is also improving. There's a third form of false positive, which is also important to keep in mind, which what happens if in the lab or part of the processing of the sample, some DNA from a different individual contaminates the DNA from this tumor. So then when we compare this DNA that has a little bit of this other person and to the normal of this individual, we will see situations like this, where we'll see a few reads in this tumor, but those reads are coming from this other individual. And of course, it's not in the normal. It's also not in the tumor of this individual. It's just in this other different person. And we will call a potential somatic mutation there. So we developed a tool called Contest to estimate, based on looking at different SNP sites, estimate the contamination level of different individuals in the sample, and then we use this quantity back in MUTECT to make sure that our model, the null model, is not that there's no mutation there, but in fact that could be a mutation there at the rate of this contamination. And that makes our tool less sensitive, but also avoid making the mistakes due to this contamination. So are there any questions so far until this point?
Questions? All right, we're gonna.
So now that we have a good mutation caller, we had a new mutation caller, then we decided to look at mutations across cancer. And together with Eric Lander and Mike Lawrence and Petar Stoyanov and Paz Polak, we studied the different cancer types. And here's what we saw across 3,000 whole exomes from 27 tumor types. Every dot here is a different patient, and the somatic mutation rate varies dramatically across individuals. And even within each tumor type, there's a huge variation. And these are different tumor types. And you see the rhabdoids and the hematologic kind of cancers like Ewing sarcoma, like leukemias, etc., have low mutation burden, whereas melanoma and lung cancer, which are due to known carcinogens, have a much higher mutation burden. This is logarithmic scale. You could see a 1,000-fold difference between the top here and the bottom. And just to give you a sense, common germline variations are roughly 1 every 1,000 bases. It's the top end of this graph. Rare germline variations are roughly, in 10 per megabase, and they're roughly at this level. And de novo germline variation between kids and their parents is roughly at this level, which is kind of the bottom of this graph. We also saw that mutations are caused by different types of mutations. So different substitutions. So for example, the melanoma has a lot of these C2T mutations, and cigarette smoke causes a lot of these C2A mutations. And but others have different signatures. And we looked at these signatures in a more detailed manner when we look not only at the type of substitution from a C to a T or a C to an A, we looked also at the base that is prior to the mutated base and one after the mutated base. For example, these all yellow mutations are C2T, but all these in these peaks are Cs that are followed by Gs. And this is what I said earlier. These are the CpG2T mutations that occur in every cancer type. And this is what's called now the aging signature, but because it's associated with a higher level of these is associated with age of diagnosis of the cancer. And this is from ovarian cancer, for example, which has this majority of the mutations are coming from this methylated cytosines that change to Ts. So there are 96 different types of mutations here. The 6 different mutations times 4 of the preceding base and 4 of the following base. When we look across cancer, we see different of these Lego plots, we call them. And you can see in lung cancer, One of the things that to identify here in lung cancer, you also see it in cervical cancer and in bladder cancer, are these spikes here. And these spikes are not noise, and they're not caused by cigarette smoke or the UV that you see kind of in melanoma. We didn't know what is that at the beginning, but then later we find out. One additional signature we found was in esophageal cancer, together with Adam Bass and Austin. We found this signature associated with gastroesophageal reflux. Now we see it also in other cancer types. This is this A2AC, which may be due to gastroesophageal reflux, but it's not clear because we see it now in other cancer types that are not related to gastric cancer, but it's not clear what exactly causes these mutations. So to analyze these different signatures, in parallel with our kind of Sanger colleagues, we use this NMF method. We looked at it in a different way. We looked at it at rates of mutations, where the Sanger colleagues looked at the counts of mutations, but it's essentially the same type of analysis, where we look at different patients and these 96 different types of mutations, and we try to deconvolve this matrix to a product of two smaller matrices that represent the different processes or signatures of mutations. And then for every patient, we could have the activity of every signature in that patient. And so we apply this non-negative matrix factorization to this matrix, and indeed identify 6 different signatures at that point. This algorithm, you actually have to tell it how many signatures to look for. We identify 6. The first one is actually a sequencing artifact that's seen in AML in TCGA. This is a smoking cigarette. This is the UV signature. This is the ApoBec signature. And this is the CpG2T kind of aging signature. And this is all the other kind of mutations. And we could cluster samples based on the activity of these 6 different signatures. And you could clearly see these are the leukemia with the artifact. Here's these samples are melanoma with a UV signature. These samples here are lung cancer. This is the smoking signatures, and the CpG2T occurs everywhere, and that's the TPC. This is kind of the ApoBec or HPV, and these occur mostly in bladder cancer and cervical cancer and some head and necks. Then the Sanger group studied 7,000 tumors and reported 21 different signatures, and then continued with version 2 with 30 signatures. And just recently, together with them and analyzing the pan-cancer analysis of whole genomes with more than 2,500 whole genomes and additional extra whole genomes and exomes, we identified roughly 49 signatures of single base substitutions, but also now we are looking at signatures of double base substitutions and indels, and you could read about this in this extra reading slide here of the recent papers. In the lab, we developed Signature Analyzer, which is a kind of a Bayesian approach for NMF. It's called the automatic relevance determination method, that basically you don't have to tell it how many signatures to look for, but it gives you something like a posterior distribution of the number of signatures. And Jae-Gil Kim developed this method. It's described in this Kassar Kim et al. And Kim et al. Here. And then we used it in different other publications from the lab. And we have now recently accelerated this method to run on GPU, and we could run things extremely fast on GPUs. But I think if you really want to understand what's the latest kind of a state of the art in signatures, you could read this most recent PCOG paper. So I will stop here for question. Ludmil is started as Sanger, now he's a PI at UCSD. Jae-Gil was in my group for many years, now we moved to industry, and Nick Ardvala was an ACB and now a PhD student in my lab. Do you have any questions so far?
How do you determine the number of the inner dimension in the non-negative matrix multiplication?
So either you supply it if you use the kind of the naive NMF or the classical NMF, but if you use this Bayesian NMF approach, and you could read about it in this paper, it actually starts with many signatures, and then in an iterative kind of process, it has some priors, of course, but in an iterative process, sees how many are relevant features that are needed to explain best the data, and you could run it from many times and different iterations, and you get kind of a distribution of numbers. So you could have, you know, there's 80%, there are 5 signatures, and or 20 signatures, and there's 10%, 19 signatures, etc. You will typically get the distributions like that. But I urge you to read more about this method that identifies, generates kind of a distribution of number of signatures, and then you could select the kind of the most likely one.
Thank you.
So now I would want to talk briefly about identifying, estimating the purity and ploidy. So as we said at the beginning, samples could have a mixture of normal cells and tumor cells, but we don't know what's the fraction in advance. So we developed a method together with Scott Carter in the lab, now with a PI at Dana-Farber, that looks at both copy number and mutation data, and looks at the allele fraction of different copy number events, or allelic copy ratio and the allele fraction of different mutations, and tries to fit kind of a histogram that is affected both by the purity and ploidy. And basically you could fit these different levels of allelic copy ratio. So this is kind of 0 allelic copy ratios, 1, 2, and 3 to the data. There's another fit that is like this, but is less likely because you see all these odd levels. Most of them don't have any copy event in them. Or you could fit this one, which is also not like likely because you are not explaining these copy ratios. So this is the most likely fit, with these mutations or these copy number changes being subclonal copy numbers, so they're in between integer copy values. So we use this method to estimate the purity and ploidy of different samples. Then we could use this, once we estimate the purity and ploidy, we could then estimate the CCF, the cancer cell fraction of every mutation. Most mutations are clonal, and this is what we call 1. But other mutations are subclonal, and since we have a discrete number of reads supporting the mutations or the copy number changes, we don't know exactly what is the CCF. We have a distribution, but for every mutation, it's a distribution. But now we leverage the fact that cluster has clones in it, and all the mutations that are shared across the clone should have the same CCF. So we apply this Bayesian clustering algorithm using a Dirichlet process, because we don't know how many clusters to expect. And using this Dirichlet process, we find the number of clusters. And what we see, this is the before clustering, and this is after clustering. And you can see it's much sharper. These mutations all share the same allele fraction, and these mutations here, and these mutations here. Using this allele fraction, we could start to build phylogenetic trees of these cancers, even from a single sample. Because you could see that these mutations that are in 70% of the cells, and these mutations roughly in 45% of the cells, the clone that has these mutations must include also a subclone that has these mutations, because 45% of the cells plus 70% of the cells is more than 100% of the cells. So there must be a subclone that has these 45% mutations that is a subclone of the 70%. So even from a single sample, we could start to do that. Build this phylogenetic trees using this what's called this pigeonhole principle. Then we extended this to 2-dimensional space, and these again are the uncertainties of single mutations of events, but we could apply this clustering in 2 dimensions, and indeed we could see that some subclones that existed in time point 1 only in roughly 10% of the cells, in a later time point increased to 75% of the cells. So these clones, we could start using these 2 time points, we could start to learn dynamics of the cancer, clones that are growing and clones that are decreasing. So together with Kathy Wu and Dan Avi-Landau and Scott Carter and Peter Stoyanov, we looked at this in CLL, and we saw that some cases showed no evolution between time point 1 and time point 2, which could be years apart, and some had clear evolution, and it was very clear that the ones that had no evolution typically were untreated between time point 1 and time point 2, and then ones that were treated between time point 1 and time point 2, clearly we saw evolution, and even the mutations in those clusters, we could identify what is the driver that causes the increase of the fitness of these cells, and therefore the clone increased over time. Now we extended this to algorithm to analyze multiple samples, because the way it was written before, it couldn't really extend. So now we have a much more efficient way to analyze multiple samples from the same patient and really understand the timing and evolution with this package that we call Phylogic NDT for n-dimensional phylogic and timing. And you could read about it. It's available in GitHub. You could read it in this bioRxiv, and it was used in two recent publications, one in CLL and one in part of this pan-cancer analysis of whole genome to study the evolution of cancer. And this package is developed, spearheaded by Ignati Leschener in the group. So I'll stop here if there are any questions. Now I want to talk about interpretation. Now that we have the characterization data across a cohort of patients, we want to find the drivers. So the way we do that, we build a statistical model of the random chance of mutations or the background rate, and then identify genes, regions, or pathways that have more mutations than you'd expect by chance. And this gives us candidate cancer genes, or could highlight artifacts or inaccuracies in our background model. So I will highlight two methods that we've developed over the years. One is GISTIC, and one is MUTSIG, or MUTSIG is a suite of tools that has different algorithms within it. And then I will also mention other more later developments. So GISTIC, in just brief, looks at copy number variations along the genome. The idea is that different samples have different gains. Different cancers have different gains. These are red stripes here, and every cancer is a column here. These are different red stripes. And these are different gains in this cartoon. And you could look across all of the samples and essentially add up those different gains. And then ask, are there regions of the genome that have more gains in this particular region than you'd expect by chance? And how do you build the model of the chance? You essentially permute these gains in every sample independently, because if there were passenger events, they could have occurred everywhere with the same probability. And so we could permute them and then generate a distribution by chance. And then convert this score, which we call the G-score, which is the sum of these amplitudes, to a p-value for every region of the genome. Then we need to correct for multiple hypotheses using kind of FDR. And those that pass this FDR threshold, we call them significant. And this is the basis of GISTIC. We now in GISTIC 2.0 distinguish between broad level gains and focal gains, or kind of arm level gains and focal gains, because they have different statistics of being generated. And that's kind of, and we have also additional dealing with peaks that are next by to each other, etc. And this is this method called GISTIC 2.0, still used by many people until today. And I'll go deeper into MUTSIG. So MUTSIG is a similar approach. You look at different patients, and these are different genes, and here are mutations. And the simplest concept of MUTSIG is just count every gene, how many mutations you saw in that gene. And then you need to compare to how many you'd expect by chance. So how do you know how you expect by chance? You could, for example, permute all these mutations along the genome randomly, and then see what is chance. Of course, longer genes would likely have more mutations than shorter genes. So for example, here are two genes. One is much shorter than this one. Therefore, the significance of these two mutations is much higher than this one. Here's one that has 3 mutations, so it's called the significance is higher. Then we could calculate p-values and correct for multiple hypotheses. We've already seen that different bases have different mutation rates depending on the signature, CpG2T, and there's this ApoBic and UV signature, etc. So we need to take the rate of different signatures into account. So we did that. We developed MutSig. And here's, again, a kind of a cartoon example of basically, imagine that you have this estimate of the background mutation rate mu, the number of mutations you'd expect to have in a gene that is size 1,500 bases across 500 tumors. Total territory will be 750,000 bases. If you assume the background mutation rate is roughly 1.5 mutations per megabase, you'd expect roughly 1 mutation on average in such a gene across the cohort of 500 patients. But if the patient is really driven by this gene, and 5% of patients have a driver there, or even 2% of patients have a driver there, then 10 patients will have a mutation. And now you can compare 10 patients with mutation compared to 1. Is it significantly different or not? And then you could calculate a p-value for every gene again, and then correct for multiple hypotheses and to generate these Q values. And if the Q value is less than 0.1, we call it a candidate cancer gene. And this method is essentially described in our paper from 2007. And we apply these tools, this MutSig, across cancer. And typically we found these cancer genes in different cancer types, more frequent and less frequent cancer genes. And what was clearly observed by them, that only a few genes are mutated more than 20% of the cases. And there's a long tail of cases that are mutated in lower and lower frequencies. But then we hit a problem. The problem was when we looked at lung cancer, roughly nearly 200 lung cancers, and running the same algorithm, we found 450 potential candidate cancer genes. And what struck us very odd is that 100 of them were olfactory receptors that are related to smelling in the nose. And many of them were very long genes in the genome, either in terms of amino acid or territory in the genome, including the CSMD1, which is, or CSMD3, which was one of the genes we thought was significant in ovarian cancer. So we didn't know what's going on. Why is the method not working? And the reason is that our method didn't take into account the fact that mutations are heterogeneous rates along the genome. If you have, we are using one estimate for a background mutation rate. And if there are regions that have a higher background rate than this estimate, then we'll have false positive there. If there are regions that have a lower background rate, then we'll have false negatives there. So we already seen that there's different frequency of mutations across different patients and across different tumor types. But also within a patient, there are differences in mutation rates in different genes. The higher the gene is expressed, the lower the mutation rate. And the reason is that there's a repair mechanism called transcription-coupled repair that repairs DNA damage in DNA as genes are being transcribed. So the higher the expression, the more chance there is to repair the damage. And therefore, you have less mutation rate. There's also another striking observation that the mutation rate is higher at late replicating parts of the genome. The genomes are not replicated from one end to the other. There are origins of replications, and there are some regions of the genome that is replicated early and some that are late. The later ones have more mutations than the early ones. Not really understand what's the reason, but it could be that they just didn't have enough time to repair. It could be that there's a depletion of nucleotides, and therefore there's more chance of misincorporating wrong nucleotides, and there are other hypotheses. CSMD3, one of our fishy genes, was indeed in a very rate replicating part of the genome. So instead of estimating a flat mutation rate, we had to estimate this kind of rugged mutation rate throughout the genome. And when we did that, we developed MutSig CV, which after we did that, instead of 450 genes, gave us 11 genes were not the genes with new one, the HLA-A. So I'll stop here to take more questions. So now with this new tool for finding cancer genes, together with Mike Lawrence and Eric Lander, we looked at nearly 5,000 tumor normal pairs from 21 tumor types and asked, Can we detect all currently known cancer genes? Can we find new ones? And have we completed the catalog of cancer genes? So overall, our database contains 3 million somatic mutations across these 5,000 cases, and 93% of the genes have at least 5 mutations in the database. So having mutation in your gene doesn't mean that it's a cancer gene. So we improved the method, not only having MutSig-CV that counted the number of mutations in the gene, we also added MutSig-CL, which looks at the clustering of mutations. If there are not many mutations, but all of them are in the same amino acid, that is unlikely by chance. And this is another indication that these are driver mutations. If another method that we added is MutSig-FN for functional impact. And we use a conservation. If there are not many mutations in a gene, but they are all in highly conserved, evolutionary conserved regions, this means that they are likely functional and therefore more likely to be drivers. So in all of these methods, it's very important to look at these QQ plots, showing that the vast majority of the genes, their mutation, their p-values are uniformly distributed, like you expect by the null hypothesis. And only the subset of the genes, there's a significant elevation of the negative log Q value or p-value. So they are significantly different from the null hypothesis. If you don't have this kind of mutation, p-values distributed along the line, then you might have a mistake in your test, and therefore most of the genes are not following the null hypothesis. Again, we believe that the vast majority of genes are not don't have driver events in cancer. So then when we apply this, here's the different tumor types. We see that we find cancer genes, and all these blue ones were previously known cancer genes, and the red ones are novel ones that we detected using our algorithm. And we ran across all these different tumor types, and we found cancer genes and novel ones, and essentially found all previously known cancer genes and in these tumor types, and added 33 new ones. So then we asked ourselves, have we completed the catalog of cancer genes? And to do that, we started to look at all the genes. We developed the tumor portal to look at the different genes and different tumor types. We could look at tumor types and see which genes are mutated in these tumor types. And today, the website looks more sophisticated, so you're welcome to go to tumorportal.org. You could look at a particular gene and see which ones are, in which tumor type it is significant and where are the mutations. This is P53, the well-known tumor suppressor gene. And here's NRAS, the known oncogene with its hotspots. And here's RB1, a known tumor suppressor genes with many mutations, deleterious mutations like frameshift and nonsense mutations scattered throughout the genome in many cancer types. Here's APC with a clear deletion or deleterious mutation, these red mutations in colorectal cancer. So indeed, we found these 33 new cancer genes. I will skip it because of time. One of them, as an example, is REB, which is a downstream of the kind of PI3 kinase AKT kind of pathway and related to mTOR. So then we asked ourselves, have we completed the catalog of all cancer genes? And to do that, we did a downsampling experiment. So for example, if you look at AML, and these are the cancer genes that we found, with 196 patients. But if we had only 100 patients, we run the algorithm, we find 19 significant genes. And if we had 50 mutations, we found 12, and 25 patients, we find 5. So we see a curve that shows us what's the trend of, if we add more patients, will we find more cancer genes? And we did it many times, and we did it for all the tumor types. And we see that all tumor types, if we add more patients, we will find more cancer genes. And also when we do the pan-cancer analysis, we will find more cancer genes as we add more cancers. Then what we did, we looked at different categories of genes. Those that are mutated in more than 20% of patients of a particular tumor type, those were essentially, we are saturating. We are not finding new of those genes mutated in more than 20%. And also the 10 to 20% is kind of starting to maybe saturate. But the 5 to 10 and below are growing and even accelerating. So we definitely see that we could find more cancer genes as we add more samples. Then we did a theoretical calculation. How many cancer patients will we need to find 90% of the cancer genes mutated at different prevalence in the population of cancer patients? So, and these are the different cancer types. The most important parameter that affects this is how many, what's the background mutation rate? What is the noise? In melanoma, there's a much higher noise, and therefore we need many more mutations, roughly 6,000 patients. In rhabdoids, the noise is very quiet. Therefore, we need only 400 to get, for example, to the 2% level, this red curve. And of course, if you have subtypes of cancer, they will split. So, breast is not one cancer type. If you split it, then you'll have fewer samples, and then this dot would go down, and we'll need more of this of each of these subtypes. So if you want to study 50 tumor types, we'll need on average, depending on the background mutation rate, we need on average 2,000 mutations per cancer type, which means 100,000 cancers altogether. We are not there yet, but this is our goal. And we're now analyzing kind of 15,000 tumors, but we could get even more exomes as now they are available throughout the world. So as we said, we will need roughly 2,000 samples per tumor type and 50 tumor types to really complete the catalog. But next we ask ourselves, what about non-coding regions of the genome? And Esther Reinbey led 2 different projects. So the TERT promoter mutation found in 2013 is kind of the poster kind of case of non-coding promoter mutations. These are in the just before in the promoter of TERT, affecting the expression level of TERT. In an effort to study breast cancer with Esther Reinbey, we've identified FOXA1 promoter mutations, and together with this pan-cancer analysis of whole genomes with the whole international community, we studied 2,500 whole genomes from different tumor types. And one of the take-home messages was that we found non-coding drivers, but less frequent than coding drivers. And roughly the ratio of 1 to 10, we expect non-coding drivers compared to coding drivers. Then other extra reading is other advanced tools that we developed is not only looking at the clustering of mutations in the one-dimensional sequence of the gene, also looking at the 3-dimensional structure. So you could have a protein that doesn't have many mutations, but all of the mutations in the protein are in the same pocket in the protein, or the same cluster in the 3-dimensional space. And that's, again, identifying a potential functional area of that protein and the cancer gene. And most recently, we found out that our models of background mutation rate are still inaccurate. And there are hotspots in cancer that are, in fact, passenger hotspots. They are not really driving cancer. And the reason that these are hotspots is because all mutational processes, there are some bases in the genome that they tend to mutate more than others. So we actually developed a much more elaborate statistical model to model this variation in background mutation rate. And using that, we have a much better map of the driver hotspot compared to these passenger hotspots. And you could read about it in Cancer Cell. So I will stop here to ask if you have more questions. So just now I want to summarize this with what is the future? Where are we going now? So TCGA and ICGC focused on primary tumors. Most of them were previously untreated. So, and that was the right thing to do, to analyze primary cancers, understanding what is the drivers, what causes cancers before we see the effect of treatment. But we haven't reached the 100,000 goal that we wanted. And in fact, the NCI has initiated some projects to try to reach to this red line in the curve that I showed earlier to reach the 2% in the population in a few cancer types. But we are still far from the 100,000 exomes that we would like to get. But there's other activities that are started and we're now pushing fast forward is striving to understand what are the drivers of resistance. So now that there's treatment given to the patients, the cancer landscape, fitness landscape is different. And mutations that were not drivers before could now become drivers because they are now conferring resistance to the therapy. So we want to understand what are the drivers of resistance. And so to do that, we're studying samples before treatment and after treatment and comparing the mutations and the clones that are growing. And these are activities that we are undertaking right now. And that's one end of the spectrum. The other end of the spectrum that is still lacking is studying kind of the pre-cancer lesions. What are the drivers that are of pre-cancer lesions, and what are the drivers of lesions in the body that will never become cancer, that are growing, they are clonal expansions, but they will never become cancer. So this is also related to what are the potential germline risk alleles, which we know some, but by far we don't know the majority of these risk alleles in cancer, and which these typically need much larger cohorts. So going forward, still we would like to complete the catalog of cancer, of drivers of primary tumors and reach this 100,000 number. But in parallel, we are going now to the early stages of cancer, understanding what are the pre-cancer lesions and what is the normal cell? What is really a clone growing of normal cells? When does it transform to cancer? What are the cells of origin? And questions like that. And then in the end of the spectrum is what drives resistance to cancer and how can we overcome resistance by potentially combination therapies that would kind of basically prevent the cells from generating these resistance mutations to all the types of the combination of therapies. So this is where we're going right now. And just to summarize the lecture, we talked about these two major tasks of cancer genome analysis. One is to characterize the genome and evolution within a patient of the cancer, and what are the mutational mechanisms in that patient. And then the interpretation task of really going across patients and asking what are the drivers, what are the subtypes, and what are the different again mechanisms and in different association with different clinical outcomes, etc. So kind of the lessons learned. First of all, cloud computing is essential for studying large data sets of cancer. Characterizations, we need to be highly sensitive and specific tools. There are clonal and subclonal mutations, and I urge you to look into this Phylogic NDT bioRxiv paper and other recent papers that used it. And then the interpretation, there's this long tail of distribution of significant mutated genes. We need to take into account the variation of mutation rate on the sample level, on the gene level, and on the base level resolution of the variation of the mutation to really model the mutations in cancer. Again, mutations can vary more than 1,000-fold across different cancer types. Mutation spectra can be decomposed into patterns to identify processes. And again, I urge you to read the paper from the PCOG on the mutational processes in cancer. Integrating multiple sources of evidence can increase the power to detect cancer genes, like the clustering and of course, the number of mutations compared to the background, the clustering, the functional effect of mutations, and the 3D clustering of mutations. We know cancer gene mutated in more than 20% of patients, but really to find the ones more than 2% of patients, we'll need 100,000 tumors. Looking at the entire genomes finds additional drivers, but not as much, but with lower, sorry for the typo here, less frequency than in coding regions, roughly the 10-to-1 ratio. And eventually, I didn't talk about experiments, but all of what we find in these statistical tools are candidate cancer genes. And then we need to actually perform the experiments to convince ourselves that these mutations really promote the growth or delay the death of cells to promote or drive cancer. So I would like to thank the many people that are involved in the studies that I described today, and also many people that are listed here or not listed here from the lab over the years, developing these different tools and different kind of analysis and projects. And again, to remind you that I'm looking for people excited to work in this area, and I'll be happy to take questions.
Hi, Gadi. For the metastatic or the resistance tumors, do you think we will be finding a lot more new mutations or existing mutations that we found in primary tumor, but with much higher allele frequency?
That's a great question. So clearly in the metastases, when we study them, maybe I'll go back to this slide. When we study a post-treatment or a metastasis or a post-treatment recurrent cancer, of course we find all the mutations that were all the way back to the fertilized egg. But if we compare it to the samples that came before the treatment, we could kind of subtract these ones. And we are looking only at the mutations that either occurred in this period of time or at least gave fitness advantage and grew, the clones grew over this period of time. And those are not many more mutations compared to the mutations that were in the entire history of the cancer. Of course, depend on the cancer type. If it's melanoma, there's more here in the history. If it's or lung, but if it's rhabdoid, it won't be as many. It also depends on the treatment. Some treatments, chemotherapies, actually generate mutations, and we can see signatures of the mutations that they generate in the post-treatment samples. Now, in terms of what are the drivers that we find, or what are the cancer genes that we find, I think we will find, and that we have some evidence of that, we'll find a mixture of two types. One are, for example, mutations that we rarely see in primary cancers that are on the target of the drive of the treatment, if it's a targeted therapy, or in the same pathway. And we find those. We also find just cancer genes that just occur later. So for example, it could be a KRAS mutation that is not related to necessarily the treatment, but it just drives the cancer faster and therefore becomes resistant to the therapy. Because if the therapy is, think of it as like putting a brakes on the car, the new drivers, which are known drivers in cancer, are just pressing the gas pedal much harder, so it actually continues to grow. And then, so I think there'll be a mixture of these two, the known drivers, but also new drivers that we've never seen in primary or rarely see in primary that are really related to the mechanism of action of the treatment.
Thank you so much. I'm going to stop